Okay, now we know that we need to produce gametes, so we need to produce these sex cells. Okay, now one important thing has to happen though before we can produce these sex cells, whether it's sperm in males or eggs in females, and that is namely that we need to reduce the amount of genetic information that these are going to contain. Okay, now typically a someone or a human will have 23 pairs of chromosomes. We have 22 autosomes, and then we have one sex chromosome. So we have 23 pairs of chromosomes. That gives us a total of 46 chromosomes. Now, if we don't do something about those numbers, that number of chromosomes is going to double every generation. So if I have 46 chromosomes and my wife has 46 chromosomes and our cells come together, well, the next generation of cells then would have 92 chromosomes. That's too many, okay? We start to run into issues of what we call dosage compensation in that case, where it's hard for us to match the levels of proteins that are being secreted or being made in cells and how to control all of that if we have that many chromosomes all at once. So we're going to try and bring that number down, okay? So what we're going to do is whenever we are making these sex cells, whether it's the eggs in females or whether it's these sperm cells in males, and when I say eggs, I'm specifically referring to what we call secondary oocytes. So oocytes meaning egg cell. But these secondary oocytes are a particular type of cell that are going to be made kind of downstream. And we'll talk about that after we are done talking about meiosis, okay? Or whenever we're talking about the female reproductive system, we'll talk about those secondary oocytes. In males, these are just sperm cells. But we need to get that genetic information reduced, okay? But we have to reduce it in a very particular way. So we can't just say, hey, we're going to take half of all the chromosomes, because we have 2 copies of every chromosome, okay? It's not just that we have 46 chromosomes and they're all the same, they all contain the same information. That's not the case at all. We have 2 copies of each chromosome, numbers 1 through 22, and then our sex chromosomes. But where our 2 copies of chromosome 1 have the same information. They just may have slightly different versions of the same information. The same thing's going to be said for chromosome 2 and 3 and 4 and so on and so on and so on and so on. Okay, so it's going to be important for us to make sure that we can have at least 1 copy of chromosome 1 get passed and have 1 copy of chromosome 2 and 1 copy of chromosome 3. All of those getting passed down into that egg cell or into that sperm cell to make sure that we have 1 copy of everything rather than just having a random assortment that somehow totals 46, okay, or 23. So the process of getting there is what we're going to call meiosis, okay? So like I said, we have 22 pairs of autosomes and we have 1 pair of sex chromosomes. The sex chromosomes are our X and our Ys, okay? Now, importantly here, these autosomes and the genes that they're going to contain, so these normal chromosomes, these are going to contain and code for information for all of our typical cell functions, whether that's cell growth, cell maintenance, protein manufacture, or a hormone being right, or maybe regulation of something like that. All of the things that our body is going to code genetic information for are going to be contained on those autosomes. So that's part of why it's so important that we have, or that we pass only 1 copy of each one, is to make sure that our cells are going to function correctly. If we pass on too many of one of those chromosomes, this can lead to problems. This is a good example of this would be Down syndrome. Down syndrome is where you get 1 extra copy or even 1 partial extra copy of chromosome 21, okay? So they call it trisomy 21, okay? Our chromosomes are also numbered in size, where the higher the number, the smaller the size. So chromosome 21 is one of the smallest chromosomes that we have. It's tiny. Doesn't contain a whole lot of information at all, but just simply having 1 extra copy of it can cause all of the problems associated with Down syndrome, or many of the problems associated with Down syndrome. And most of that is going to be due to what we call dosage regulation, which is making sure that we're getting the right amount coming from each of the copies. Because we have 3 copies there, all of a sudden, those ratios are not going to be off and things aren't going to work the ways they're supposed to, which can lead to, like I said, the problems associated with Down syndrome. But these are going to be important for maintaining typical cell functions and giving us all of our characteristics or many of our characteristics. Now, sex chromosomes are a little bit different. Sex chromosomes, the X and Y, instead of being the same, or what we call homologous like there are in the other ones, like in the autosomes, instead they're going to be different. The genes that are encoded on the X chromosome and the Y chromosome are quite different, okay? Like very different. The Y chromosome has lost a lot of its gene functions and only has a couple of genes that are found on it. However, if you have a Y chromosome, you're typically going to be male. If you have an X, or if you have two X chromosomes, you are going to be female. However, there are variations where this doesn't actually happen, where one of the chromosomes that does not get passed on, like passed into the sex cell that gets fertilized and stuff like that, or does the fertilizing, maybe it's missing one of these X or Y chromosomes. So you get an X from, say, the mother, and then you don't get anything at all from the father, and all of a sudden you have someone that just has one X and then kind of an empty space, what we call an X naught. That actually does happen fairly often. In this case, they are going to present mostly as female. However, some of their characteristics may be slightly off, and that's just because they don't have this double dose of some of the things that are coded on that or with that double X chromosome. Sometimes things also happen where you get someone that is XXY, where they actually get two copies of the X chromosome and then one Y chromosome. At this point, that is also going to cause its own issues. So don't think that we are only related to or only stuck in this kind of XX or XY process, okay? But all of that leads us to this next part, which is to say we are going to be trying to go from a diploid cell where we have 23 pairs of chromosomes down to a haploid cell where we have 23 chromosomes. We have one copy of chromosome 2, or one copy of chromosome 2, one copy of chromosome 3, and so on and so on and so on and so on, and then one sex chromosome. That is it. This is to make sure that we don't get two sets of everything. This way, we're not constantly doubling our chromosome size every generation. Because at some point, you would also run into a case of you simply have too much genetic information and you can't replicate your cells fast enough. Okay. So what we're going to do is we're going to start with this diploid parent cell. Then we're going to produce two haploid daughter cells. Okay. Or sorry, produce four haploid daughter cells. So we're going to start with a diploid parent cell and then end up with four daughter cells, all of which are haploid. That's to say they all contain only one set of every chromosome. So they're going to have one copy of every chromosome, and that's it. Okay. Now, this is, of course, going to be happening in the gonads, so in the ovaries of a female or in the testes of a male. And this process that we're going to undergo in order to get us here is what we're going to call meiosis. Okay. So normal cell division, though, you start with a parent cell and you end up with two daughter cells that are genetically identical. No differences. Okay. So start with one, you just replicate everything. You end up with two cells at the end of it, and those two cells are exactly the same as the parent cell. Okay. And it just is a single division. In meiosis, it's a little different. In meiosis, this is only for sex cells. We're going to start with one cell. We're going to divide twice. We're going to start with one, go down to two, and then we're going to divide those two, and we're going to divide down into four cells. Each of those four daughter cells, though, is also now going to be different from the parent cell. Why? Because they're going to contain half of the genetic information each of the parent cell. Okay. But if you say take four halves, that still leads you to two, which means we're going to have to replicate the DNA as well. Okay. So just like mitosis, we will actually have to replicate all of the DNA. It's just we're going to replicate the DNA and then we're going to start dividing it and dispersing it down towards the daughter cells. Now, one other interesting event is going to happen here, and that's going to be called crossing over. Crossing over is where homologous chromosomes, so say our chromosome 1s, where they can actually exchange information. Okay, so like I said, we all have two copies of chromosome 1. We have a maternal chromosome 1 that we got from mom, and we have a paternal chromosome 1 that we got from dad. Now, because we have this chromosome 1 that we got from mom and chromosome 1 that we got from dad, we'll say kind of draw it in blue and red. So here's chromosome 1. Yeah, it's fine. They'll just be bigger, so it's easier to see. So here's our paternal chromosome 1, and here is our maternal chromosome 2, okay? Or our maternal chromosome 1. So each of these is chromosome 1. So in the event of crossing over, what's going to happen is they're quite literally going to cross each other over. So they are going to swap some amount of DNA between them, which is going to lead to to our cells having a combination, or our chromosomes having a combination of information of chromosome 1. And so be partially maternal and partially paternal. So now our chromosomes are going to have not just maternal or paternal DNA. Now they're going to have a combination of both maternal and paternal DNA. Now, this is only going to happen in one of our pairs, okay? So say we started like we have a normal cell. Our first part of meiosis is actually going to be the replication of DNA. Well, if we start with 1 and we replicate it, what are we going to end up with? We're going to end up with 2. So then we're going to have 2 copies of chromosome 1 or 2 copies of maternal chromosome 1 and 2 copies of paternal chromosome 1. Well, the crossing over event is only going to happen between 2 of these. Okay, so we're still going to have a full-on maternal chromosome and a full paternal chromosome as well in our first set of meiosis. Now, then in our first division of meiosis, what all we're going to do is we're going to take this and then divide these into 2 separate cells, okay? So we'll have a daughter cell here and a daughter cell here. The next step of meiosis will actually be to further divide these, where we then get 4 individual cells, where then we just randomly assign one of these chromosome copies to each one of these. So then we're just going to kind of pass them around, where we'll have some that will have a paternal. Hopefully you can see this. I don't know what it's looking. I'll move this so maybe you can see it a little better. Okay. Where we're going to have our paternal get passed. We'll also pass the maternal to 1. But then the other cells will have mixtures. Okay, so some we'll get those kind of partial or mixed chromosomes, where we're passing on both maternal and paternal genes at the same time. Okay, that's the goal. Now, this is going to happen not just for chromosome 1, but this will also happen for chromosome 2, 3, 4, 5, and so on and so on and so on and so on. So each individual daughter cell is going to basically get randomly assigned either a paternal chromosome, a maternal chromosome, or a mixed chromosome for chromosomes 1, 2, 3, 4, and 5, and so on and so on and so on and so on. Okay? Now, eyeballs. I've been asked several times if there is any chance at all if you can pass on only DNA or only information that you got from your mother. And the answer is that it is, I mean, I guess technically possible, but it would be very, very, very unlikely because, like I said, this is randomly distributed, okay? Now, because it is randomly distributed or randomly assigned, and this is kind of a law even in biology, because it is randomly assigned, the odds that this would occur would basically be, well, let's see, what are the chances of any one particular gamete over there getting just this paternal chromosome? Well, it's 1 in 4, right? So if it's 1 in 4, you would have to do that for each chromosome, right? So you would put 1 in 4, and then you'd multiply that out by the total number of chromosomes, which would be 22. This is an astronomically large number, okay? And by that, I mean close to the number of stars. So technically possible has likely happened, especially if you think about how many gametes males in particular make. So it has likely happened, but it is incredibly unlikely. Okay. Just because that number is, like I said, just phenomenally large. But I guess technically possible. Now, the reason that we do this and the benefit of going through all these processes is to once again make sure that the daughter cells or that the growing embryo has only 2 copies of every chromosome, just the way the parents had, so that we're not doubling DNA size every generation. But also, by having these 4 different options that we're getting out of our gametes, this is also ensuring that there is a mixture of DNA happening into the next generation so that we can generate additional genetic variation, which would maybe have a better chance of surviving than our current generation. So this is going to give us, like I said, additional variations and increasing our genetic diversity, which is something that is a little bit helpful. Now, what I drew is basically going to be how meiosis is going to work. Now, this is going to happen basically after our cells are growing in this phase that we call interphase. The homologous pairs of chromosomes separate when the cell divides, okay? So these homologous pairs, okay, so our chromosome 1s, they will separate as the cell divides. So this is going to result in 2 cells. So meiosis 1, let's just skip through the phases. I'm not worried about the phases too much. Okay, so we replicate the material, and then we're going to divide, okay? After we divide, we basically end up with a set of daughter cells, which are going to contain a duplicated chromosome 1, 2, 3, 4, 5, and 6, and so on and so on and so on, where each of the daughter cells are going to have 2 copies of each of these, okay? Now, the daughter cells, though, are going to contain at least chromosomes that are somewhat related. So they're either going to have a paternal chromosome, and then connected to that paternal chromosome will be a mixed chromosome. They'll receive a maternal chromosome and a mixed maternal chromosome. So that's how they're going to divide, okay? The second stage of meiosis is actually going to be that second division where we're going to take those replicated or those stuck together homologs and then basically just dividing them out into their daughter cells, okay? Or these haploid cells where we're only going to get 1 of each, okay? Now, just like we saw before, we're going to have the cells line up in the middle. Where the cells, or when the chromosomes line up in the middle of the cells, we're going to call this metaphase, where meta means middle, okay? That's kind of your big clue that you are in metaphase is when you see them lining up. To the point where when this happens, we oftentimes call this a metaphase plate because you can see it and almost looks like there's this line in the middle of the cells that are helping this occur. Now, if you've ever kind of wondered what the centrioles in our cells are doing, our centrioles are actually here to help make sure that this process happens correctly. The centrioles are here to make sure that the chromosomes line up correctly on that metaphase plate. And then they are going to pull them apart and make sure that the daughter cells are going to receive the correct number of chromosomes so that we don't end up with these situations of having too few in some cells or too many in other cells, and especially in these sex cells because of all the problems and stuff that can create. Say, for instance, that you get, like we talked about how Down syndrome is because you get 1 extra copy of chromosome 21. Well, that's because in this second division, it just pulled the entire pair of homologs, okay, into 1 set of cells. That can be a problem. Other problems can be created if you don't receive a chromosome. Now, like I said, chromosome 21 is one of the smaller ones that we have, but say you do that with chromosome 1. All of a sudden, you get 3 copies of chromosome 1. Well, if you get 3 copies of chromosome 1 and somehow that cell gets fertilized or ends up in an embryo, now they have 3 copies of chromosome 1, that embryo will likely not make it very long. Like in the case of chromosome 1, maybe not even days. Just because through the process of cell division, the cell realizes that it's not working correctly and then will no longer replicate or the cell will die just simply because there's way too much of all of those chemicals or something like that. and it will cause massive amounts of problems. Only in cases of very small chromosomes like 21 does it make it all the way to term and for someone that can grow and live an adult life and things like that. For anything larger than that, it's so catastrophic that they usually don't make it even full term. Sometimes they will pass away before that, or will be reabsorbed or something like that well before that. And this process or this problem is what we would call non-disjunction. These are, like I said, can be catastrophic in some cases. So most, but not all, trisomies and monosomies are lethal. Okay, so that is how meiosis is going to work.